Welcome, everybody. This is the Rotary E-Club of Silicon Valley, and every week we bring you the stories of people who are making the world better. They do this locally, globally, and digitally. We have about 40-something members in a dozen different countries. We're part of Rotary International, over 1 million members and 36,000 clubs, and these clubs come in all shapes and sizes. Ours is an online and asynchronous club. What that means is that we have people who have a heart for serving others, but are crazy busy in their schedules and need something a little different. And they might really like that global focus that we bring to the things that happen within Rotary. We are excited to be telling the story of Dr. Dave Duffy's work at the University of Florida Whitney Laboratory and Sea Turtle Hospital, where he has done things that got us very close to the words Star Trek tricorder, which made me think this is going to be a truly awesome presentation. Dave, it is such a pleasure to have you. Welcome to the Rotary E-Club of Silicon Valley.
Thanks, Rustin, and thanks for inviting me to talk to you today. So as you said, I'm going to talk to you today a little bit about Star Trek maybe, but mostly about airborne biodiversity genomics. It's a bit of a mouthful, but I'll explain what that is as we go along. But essentially, it's a new technology for monitoring nature on a global scale, both looking at wildlife and their pathogens as well. And just kind of to get us grounded in terms of Rotary alignment with Rotary, there's probably three main Rotary areas that this talk will touch on. The first and maybe most obvious one is protecting the environment. The second is providing clean water, sanitation and hygiene. And the last one is fighting disease. So at the end, I'll come back and point out how the aspects of the talk relate to each of these three areas. Okay. And also in trees, my lab has three main research pillars. So I'm only going to talk to you about one of these today, which is environmental DNA. And I'll explain what that is shortly. But just so you're aware, we also work very heavily on sea turtle diseases, especially tumor disease, and on plastic ingestion as well. So if you ever have interest in that in the future, feel free to ask me questions, or maybe I can talk to you about that in the future at some point as well. But for today, I'm going to focus on our work on environmental DNA. And we use this emerging technology to look at specific wildlife species of interest, such as sea turtles. We also have a human element. We use it to look at wildlife and human pathogens. And essentially, one of the big interest areas here for us is its ability to look at everything, all of biodiversity, in one snapshot. And essentially, what we do a lot of is try to push the limits of what eDNA technology is possible for. Always looking to the future as to what extra information can this technology provide us. And before I show you any of our work or any results, I just want to acknowledge that it really does take a village, that everything I show you, there's a huge team of dedicated people behind it, everything from students, postdocs, staff, interns. We have citizen scientist teams working with us, and many international collaborators as well. So just know that everything I show you isn't in isolation. It's thanks to the work of all these fun people and many more that we even have these results to share with you today. And essentially the problem, you've probably all heard, but biodiversity is being lost at an unprecedented rate. So even since the 1970s, global wildlife has declined by a shocking 69%. And there's huge implications for nature. And we're not just losing individuals, we're also losing a lot of genetic diversity in these populations as well, which has knock-on implications for how susceptible they are to diseases, has knock-on implications for us in terms of the emergence of new diseases and pandemics that can transfer from animals to humans. And even selfishly thinking from a human point of view, it also limits our ability to discover new drugs in the future because many of our drugs actually come from nature. So this is a huge problem globally. Not just for nature, but even for the most selfish of humans, this is going to have impacts on them as well. And this decline is happening across all ecosystems. So unfortunately, there's no ecosystem on Earth that's not impacted by these biodiversity declines. The most heavily impacted is freshwater, largely because that's where humans are, is next to freshwater habitats, so we're impacting those at a higher rate than the other environments, but all environments are being affected. And while this wildlife is declining, the amount of humans and the amount of our companion animals is actually increasing. So wild mammals now only make up the weight of 6% of all mammals on the planet are actually wild, with the remaining 94% actually being us humans and our companion animals and our domestic animals as well. So there's been a shocking change throughout the human era in terms of a concentration of species where we're losing all of this diversity of wildlife and it's concentrating down and down into ourselves and domesticated animals. And thankfully, there's a lot of initiatives, including global initiatives, to try and tackle biodiversity decline. But this is a big problem. It takes a lot of targeted investment of effort and resources. And up until now, it's been really difficult to quantify, well, actually, which efforts are working. So if you want to mitigate biodiversity, you want to restore it, you have to work from the local all the way up to the global scales, and you have to work across ecosystems and species. So having easy ways to actually monitor if what you're doing is being successful, if it's really restoring the most amount of biodiversity possible, has been really problematic. It's difficult and time consuming and it requires a lot of experts. And at the same time, with all this loss, we also want to recover genetic diversity before it's lost. So how can we kind of improve measurements of biodiversity while also recovering this genetic diversity information? And essentially what that needs is novel technologies. And one of these technologies which we think can play a really large role here is known as environmental DNA genomics. So you've heard me mention environmental DNA before a few times now. And what it essentially is, the easiest way to think about it is forensics for wildlife. So all species, including ourselves, are constantly shedding minute amounts of our DNA into the environment. So wherever you're sitting right now, that environment actually has some of your DNA in there. And this happens through a variety of mechanisms, including the shedding of skin, the shedding of hair, bodily fluids and excretions, but also every time we exhale as well, we're leaving minute amounts of our DNA behind in the environment. And this is true of every single organism. Every animal, every organism is leaving telltale traces of DNA behind. And essentially what environmental DNA is, it's recovering this DNA from the environment. So whether that's from water samples, whether it's from sand and sediment samples, we now have the technologies where we can recover and purify that DNA from those environmental samples. And once it's been recovered, we can work with it just as we could DNA from any other conventional sample, like a tissue sample or a blood sample. So that means we can actually study the animals that have been present in the environment without ever having to see them, without ever having to interact with them. This means it's a really great tool for monitoring difficult to reach or cryptic species. It's also non-invasive, so we can study these animals without ever having to be present at exactly the same time, without having to observe them, but also without having to cause stress to them if you're thinking about wildlife populations that are vulnerable. And at the same time as recovering this DNA from wildlife and plants, you're also recovering DNA from pathogens as well. So essentially what my group tries to do is to take advances in genome sequencing technologies, which are largely driven by the human medical field, and apply that to these sort of environmental samples to allow us to understand more than we ever could before about wildlife and biodiversity. And in recent years, our group and other groups around the world are increasingly now also using air as an environmental medium, where we can also recover DNA from the air and use it for all of these downstream applications. So, as you alluded to at the start, this is actually taking us a step closer to the Star Trek tricorder. So a real ability to scan an environment and have an idea of what's present, be it plant, animal, pathogen, or fungus. And we're not there yet. This is still kind of technology that's under development. And we're a lot closer than we ever, I think, expected to be at this point. So while we can't be as instantaneous as they can in Star Trek, give us a few years maybe, we can take an air sample and we can go from that air sample all the way through to an analyzed biodiversity and pathogen list in less than 24 hours. So we're getting closer. And some of the reasons we're able to do this are because we can use cloud-based computing. We can upload samples from remote field sites, even over older 4G cellular networks. And we have technologies that are portable that you can take out into the field and sequence DNA on site practically anywhere in the world. And the combination of these technologies means that we can do all of this sort of analysis quite rapidly. And with the advent of AI and other advances in computational power, we think these times are going to continue to drop and drop once these technologies are developed. And we're already at the handheld portion of a tricorder. So sequencing devices used to fill entire warehouses about 20 years ago. Now they're down to the size of something that can fit in the palm of your hand. So Oxford Nanopore is one company that develops these devices. There are others, but essentially this portability has already been achieved and costs are continuing to come down. So we can again see in the future where scanning environments to understand what's present is going to be only more achievable in shorter timeframes. So the study that I'm mostly going to speak to you about today was essentially taking these sequencing technologies, both these portable ones and other ones that we have, larger ones in the lab, and applying it to airborne DNA that we pull from a whole variety of environments and comparing that with other types of eDNA samples as well. And essentially, by doing this, you can monitor for all sorts of species just through taking air samples alone. And like I said, these samples can come from almost any environment on the planet where we can pull air, we can sample air, we can recover the DNA from that air, and then we can see what species are present. And the amazing thing about this is at the same time, in these relatively quick timeframes, we can detect the DNA from thousands of species in the air, from viruses all the way through to vertebrates. And this has a lot of implications for health, wildlife, and biodiversity. And the really important thing here is that simultaneously you can recover lots of information that's actually valuable for lots of different issues facing society as well. So this isn't even an exhaustive list, but I won't go through the whole thing. But at the same time, you're recovering information for things such as that can be used for pathogen genomic surveillance. We can use it to look for antimicrobial resistance. We can use it to look for disease vectors, to look for allergens in the air, but also to conduct these rapid, broad-scale biodiversity assessments where you're not just looking at a handful of species and trying to infer from that. You can really look at the amount of DNA coming from all of the species in that sample. So on the right are just some example images of species that we've been able to pull the DNA from the air and assess it with these rapid simultaneous approaches. And essentially what we found is that outdoor air does indeed carry high quality DNA. That means we can do a lot of downstream analysis. I won't go into the details, but essentially we've replicated a lot of this at study sites in Florida in the US and also in Ireland. And we can compare between these sites and across time as well. So just to give you an example of how much information is actually floating in the air, this is results for human DNA in one forest sample site in Florida. This forest is mixed use. It's got some residential, low development residential areas as well. And from pulling air from that location, we can detect the level of human eDNA that's present, but we can go deeper. We can look at the diversity of the DNA present. So for instance, here in this one sample, we're able to find 8 different human haplogroups, so genetic ancestry from 8 different areas just from this air sample alone. And if you take this process and do exactly the same thing in a city center, you recover more human DNA, but you also recover more diverse human DNA. So in this case, in July in 2023, in Dublin city centre in Ireland, we could recover 87 different haplogroups. And the following year, in 2024, we again got a lot. We got 84 different haplogroups. So this means we can learn something not just about the species that are present, but also about their genetic diversity, their origin. You can do this not just for ancestry, but also for susceptibility to diseases and any other sort of information that's carried on the genome. But as we started talking about biodiversity, it's not just humans. So in this very same sample where we can go and look at this genetic ancestry, you're actually able to see what other species are present. And you can detect thousands of species, everything from microbes all the way up to vertebrates. So we get things like bird species and bat species. We'll get insect species such as mosquitoes and butterflies. You can get fungal, you can get plant DNA as well, reptiles such as snakes and alligators. So again, just from surveying the area, you can get a really good understanding of what species are present and the relative abundance of their DNA in each air sample. But this is really rich information. So in this single sample, we can zoom in on any one of these species. So in this instance, we can look at lynx or bobcat. And okay, we know how much DNA is present from that species in that sample, but we can also do the same sort of genetic ancestry analysis. And in this case, this was a sample taken in the Florida forest. And when we analyze the DNA from the bobcats that are present, we can see that it matches other bobcat DNA samples known to come from Florida. So it's telling us that this DNA is really still containing the original information that it would have had when it was attached to the animal. And this is really important for migratory species such as birds and sea turtles, because to protect the species, it's not enough to know just where they are. You also want to know where they come from, because they need to be protected across all of the different areas that they use. And at the same time, we're pulling information about viruses and plants and lots of their species as well. So this is really, really rich data sets that can be put to a huge variety of uses. Just to give you an indication, it's not just mammals and large animals. You can do this for invertebrates. This is a golden orb weaver spider. Again, we can see that they're present in the environment, and we can tell that they indeed match with other known Florida sequences. So this is grant rooting out and giving us confidence. But you can think of applying this for biodiversity assessments, for trying to detect invasive species and knowing where they came from, and also importantly for insect vectors of diseases. So what's known as the most dangerous animal in the world really is the mosquito, because it's responsible for over 1 million deaths every year and up to 700 million human infections. So it's a hugely problematic species for humans and for animals too. But it can be very hard to monitor. There's a lot of different species, and a lot of them actually don't respond to the mosquito traps that we have. You need different traps for different species. But just like the spider, we could actually see that we are pulling mosquito DNA out of the air as well. So we could now see differences. Instead of looking at one sample, you can compare between your samples. And we see differences in different locations. We see differences in the same locations over time, and that we can detect multiple different types of species as well. So actually, while we can do these broad-scale biodiversity studies from the air, we can also hone down on species of interest and really get an understanding of what's happening with those, and if the mitigation efforts we have against them are being successful or not as well. And this works even for species that are smaller than mosquitoes, would you believe? So even biting midges that we have here in Florida as well, known as no-see-ums, they're really tiny. You can see them here in a human arm next to some hair. We can detect these over time as well, and we can see differences between different sites. And again, this tallies with what we know about these sites in the open areas where our research institute is. We tend to see a lot more of these than in the forest locations, and that was borne out by the eDNA analysis as well. So this is a really powerful tool for investigating species that are disease vectors. Again, from a human point of view, we can also look into these very same samples and look for allergens such as peanuts or pollen, fungal spores, dust mites, and other food items that are of issue to humans. This works indoors and outdoors. We can pull all sorts of types of information from these samples. And we can also pull information about species that are linked. So in this case, hosts and their parasites. We can find out the level of eDNA in the air, for example, here, bee species. We can see how that changes over different sites. At the same time, we can see how much DNA is present from their parasites, which causes a lot of problems for bee colonies. So we can see how we can study host-parasite interactions, but you can also be studying things like pollinators and their abundance and the effects on pollinators, which are crucial for our own crops as well. And just to point out, it's a little bit strange, but this sample that had the highest amount of bDNA was actually our city centre site. And that links back to the fact that sampling was done at a university, which has a well-established campus pollinator plan. So again, if you're measuring biodiversity mitigation and enrichment efforts. You can see here that it looks like the one for pollinators is working. And just to point out, I won't go into this in detail, but you don't have to suction DNA directly from the air. DNA is constantly settling as well, so you can actually go out and sample DNA just by swabbing surfaces. In this case, you can swab a window pane and you get DNA for all these species and viruses as well. So I'll just take a pause for a second to say this is a really simple approach, but it can be very, very impactful. So this is an example of a filter where we captured DNA for a week from the air in Dublin city centre, and I hope you can see the difference that the original filters are quite clean and pristine looking, whereas after sampling you capture a lot of pollution, but in that filter we have DNA for all of these species as well. And really the only limit is the human imagination for what these samples can be used for, what they can be analyzed for. And we do find, depending who we're talking to, different people get very excited about different components of the results. Everything from narcotic surveillance through to disease and pathogens. And people were really excited to know how much dog DNA was in the air in Dublin as well. So depending on your issue, there's absolutely applications that can be enhanced by airborne DNA analysis. And just to highlight that these approaches work equally well, not just for air, but actually from sand and sediment and water. And sand, sediment, and water are samples that are traditionally have been examined for eDNA for longer, but by taking these same approaches, we can do the same thing, whereas instead of looking for an individual species, we can actually look across all of the species that have left telltale signatures in those samples too. And I'm nearly out of time, so I'm not going to labor the fact, but just to point out that there's huge amounts of information can be gleaned from the sand. So a lot of our earlier studies, just from taking tracks on a beach, you can tell what species of sea turtle made those tracks. You can look to see what pathogens that mother was shedding as she came ashore. And we can actually identify the natal origin of that mother as well. So where in the world she originated from. We have a really extensive sea turtle beach monitoring program now, which is only possible because eDNA is so simple to take the samples. We have a huge network of dedicated citizen scientists who are able to recover samples from all over the region and send them to us. And this really doesn't take much. So from beach sand, 10 grams of sand, even a quarter of a gram of sand is enough to recover sea turtle DNA from as well. And just from this sand alone, we can identify exactly where that animal has come from, which, like I said earlier, it's really important for migratory species to know not just where they are, but where they have been, so that you can protect them across their entire range. Just protecting them in one location isn't sufficient, and it's particularly important for species like sea turtles that cross national boundaries as well. So we know some of our turtles here in Florida are coming from as far away as Brazil. And again, I'm not going to go into these details, but just to point out, these same sort of biodiversity assessments can be done from water as well. You can get all sorts of thousands of species, from viruses to vertebrates again. You actually get signatures for terrestrial species because their DNA will eventually wash into the rivers, and you can detect them there. This works in seawater as well. And again, humans are animals, so we're also shedding DNA. So by monitoring water samples, we can actually point source where wastewater is incorrectly entering water systems. So we can look for human DNA and we can see where improperly treated wastewater is. If mitigation efforts happen, you can see has that actually solved the problem. So we have samples from sites where we can see before and after proper wastewater, and we can see large human signals while the wastewater is entering, which disappear once proper sanitary measures have been put into place. And because we're getting information, everything simultaneously, it's not just looking at humans in those very same samples. We can look for viruses and pathogens. And again, where we see high levels of human DNA indicative of improperly treated wastewater entering that system, we can also find pathogen DNA from lots of known human pathogens as well. So we're not naive to the potential implications for human environmental DNA studies as well. And we put a lot of work. We published a large paper back in 2023 where we discussed both the benefits of human eDNA research, but also potential ethical implications of it as well. And we were able to get a lot of regulator and media attention in this issue so that we could maybe have more nuanced regulations moving forward, both where and when these approaches should be used. And just to point out that we've shown that these approaches from both sand, water, and from indoor and outdoor air as well, all recover human DNA, which contains a lot of information, including disease susceptibility information. So there's a lot of ethical considerations here as well. But essentially, the take-home message is that environmental DNA technologies have huge potential to continue to develop at pace. They will be routine tools for biodiversity monitoring, for pathogen surveillance, and also for conservation managers as well. Every single window pane in the world is actually acting as an error eDNA sampler. You can go along, if you just don't clean your windows too regularly, you can go along and recover that DNA and see what species are in the area. And there really is a lot of information, just 10 grams of sand, 500 mils of water, or thin air itself. And just because I promised we would come back to the Rotary specific alignments. So eDNA technologies are proven, but they still need more research and they're still continuing to advance. And in terms of protecting the environment, I hope I've convinced you that they're really great tools for rapid biodiversity assessments and truly biodiversity. Not just picking a couple of species, but actually seeing everything from virus to vertebrates and seeing if what you're doing to try and mitigate biodiversity loss and to restore biodiversity is really helping a few species or if it's helping across the whole breadth of biodiversity in that area. And they can also be used then on a more individual basis to track wildlife and identify their origin as well. In terms of providing clean water and sanitation, so these are great tools to identify the source of water contamination, whether it's improperly treated wastewater coming from pipes or whether it's septic tank leaching, or indeed whether it's other sources as well. Not only can you see how much of each species is there, but you can tell, okay, does it look like it's human wastewater or does it look like it's agricultural runoff? So if you can identify what the problem is, it's easier to then mitigate it and clean it up. And at the same time, these technologies can be used to assess the level of pathogens and parasites in water and to know if they're actually safe to drink or not, or if there's extra measures that might be needed. And then finally, in relation to fighting diseases, we can do this simultaneously or we can do it in a targeted approach, but you can use environmental DNA to monitor for pathogens, to monitor for parasites, to monitor for allergens as well. And you can also use it to monitor for disease vectors such as mosquitoes and other insect vectors of diseases. So again, just to highlight, all of this research is still ongoing, but there's huge potential. And in order to achieve the full potential of eDNA for both societies and for nature and biodiversity, further investment in research is absolutely required. And that's why we're also more than happy to discuss with potential collaborators or donors, sampling in your favorite habitats or applying these technologies to issues of concern to you or to your area as well. With that, I'd just like to thank the various funding bodies that we've had throughout the years that have supported all of the research that's conducted in the lab, including the eDNA component. And again, finish where I started, acknowledging that this is really a culmination of the work of a lot of people, a lot of students and postdocs as well, and particularly for the Air eDNA study, I should acknowledge Orestes, Mark, and Max, who are heavily involved in this. And with that, I will take any questions you might have.
Well, I think we can get a few in. Thank you very much. If you would stop sharing for a minute, I'll introduce the people that we've got in the group. From our club on Catalina Island in Southern California is Ernie. In Vancouver in British Columbia is Nick. My name is Rushton. I'm in the San Francisco Bay Area. Dave, how might groups begin to accelerate the kind of work that citizen scientists have done as a part of this research? So, and let me add to that question. Are the tools for doing this kind of environmental DNA analysis the kind of things that can be in the hands of just a few people in groups here, there, and anywhere?
Yeah, so one of the big leaps that kind of has to happen in terms of sampling, we've reached the citizen scientist stage where there's lots of great projects around the world where you have citizens who can take samples. Generally, the lab analysis still happens in a smaller number of labs. There's many labs doing this, but it's not generally done by citizens yet. And the real bottleneck is the computational analysis. So I didn't go into specifics, but there's different ways you can do environmental DNA and assess the DNA. You can look at individual species or pathogens, or you can go all the way up to these whole biodiversity assessments. And even within that, there's different ways of doing it. But the ones that really give you the most amount of information, where you're really getting information from everything that was present in the sample, that's computationally very intensive. So it's not currently easy for that to be done by citizens or conservation managers. But it is something that, as it is established by researchers, as we understand the best ways for analyzing that, it is possible to then make computational pipelines that could actually be implemented by people more broadly. And we do see AI as having a big part in this in the future, as rapidly being able to figure out the best pipelines. There's always a balance between the maximum amount of information and also being sure that the information you're generating is specific as well. So there's thresholds. It's not a clear yes or no, and there can be differences in sequences between populations from different areas as well.
Good deal. Nick, I'll pass it over to you.
Yeah, and thanks, Russian, because you asked one of the first questions I had, which is how we can get involved. And I look forward to the time when we can, because it seems like something that, especially teaching those around us, like our kids, to be part of this would be really cool. I guess I'm curious about the evolution of the technology, and specifically how this is kind of combined. I think of how COVID influenced our understanding of airborne illness. All the people saying it's, how the airflow needs to be in the offices and stuff, but also the kind of commercial and medical sector. How are they influencing this work? Because I see, like, obviously, hopefully they're providing a lot of that funding, like you highlighted, but a lot of the science too is being applied there. Is that where it's going?
It is as well. So there's a convergence, but also just to recognize that the technologies that we rely on to do all of this DNA sequencing generally are driven from the human medical field. So the real advances, reductions in cost, most of that happens because those technologies have medical applications. So we kind of benefit from that. In terms of directly benefiting each other, COVID accelerated as well the use of environmental sampling. So it's a bit technical, but there's delineations and people argue over when environmental DNA started. Some people are doing it for microbes, and you can take swabs for microbes a long time ago. Some people only say that when it's for animals, that's when it starts to be environmental DNA versus microbiology. From our point of view, that's kind of an academic argument. What we're interested in is how is this beneficial going forward? And actually, by looking at everything together, there's extra benefits there. So with COVID happening, that really accelerated the push for wastewater monitoring for diseases and pathogens. So there's some really great advances in monitoring city wastewater systems, and actually for COVID and now subsequently for other pathogens as well. You actually get a warning of about 2 weeks before you start to see hospitalizations for COVID, that you can see a spike in infections. You can find out what strains are present just from monitoring wastewater. So a lot of countries now have developed these monitoring systems where they're routinely sampling wastewater and can see what's coming down the line, essentially, in terms of pandemics, but also other endemic diseases as well. So that's probably where environmental approaches have been most applied, but it's also getting a lot of traction for things like bird flu as well now, where you can go out into the wild and you can recover viral samples from water or from our sampling in other locations as well. So I think there is this convergence where it's being recognized that a lot of the places where you really need to be if you're a virologist or a microbiologist looking for that next agent that could cause a pandemic in humans or could cause serious diseases in wildlife or farm animals. Most of those interactions are happening in hard to reach places. So actually, environmental DNA is really well suited as a field technique to be able to be applied to those places and gain a lot of insight. And you don't have to wait until it's already in the patient population.
Let me ask about several different use cases for this. So one of the things you talk about is allergens. I mean, can you imagine someone who deals with severe allergies using tools like this in order to really monitor their environment for the kinds of things that might be dangerous to them?
So in the future, yes. Because we know these things are not carried on the air as well, you can do these computationally intensive approaches, which tells you everything that's present, but other people are developing tests more akin to lateral flow tests that you can take with you. And, these are being developed for, say, fish species. You can go to a water body and you can just test the water, put it on one of these things in the field, and you get a positive or negative. It doesn't tell you how much, and you can't do the subsequent DNA analysis, but it certainly tells you positive or negative. So you could see how those sort of technologies could be readily applied to single-use tests of people. If you're, I don't know, going to enter in a closed space like an airplane, you could monitor this sort of thing. Equally, people are getting inventive, and you can pull DNA out of the air really easily. A lot of our HVAC systems are already doing it. Anything with a filter is actually capturing it. So you can use those filters to test retrospectively what was actually in that air as well. So I think in the future, as those things get quicker and easier and more cost effective, there are absolutely applications there as well that would be beneficial.
Okay. I've read about programs where genetically modified mosquitoes have been released into environments in order to try to overwhelm the natural mosquito population in order to limit the kind of disease transmission that might happen. Will this kind of technology accelerate efforts like that?
Yeah, so what maybe where it can have the biggest impact is mosquitoes. You can also have invasive mosquito species which have been introduced by humans in new areas and they bring new diseases. So monitoring those becomes easier with these technologies. Because you can target different regions of the genome, you can also potentially use these approaches to look not just what species are present, but you can look for resistance markers as well. So a lot of mosquito species are becoming resistant to the chemical compounds are used to try and minimize their numbers. And again, that resistance, it's genetic, it's inheritable, so it can spread through populations. So as well as surveying for species, you can actually survey for the spread of these genetic markers. So instances like that can tell you, oh, okay, it looks like this resistance is spreading and you might need to change up what chemicals you're using. Equally, if you can get it sensitive enough in the future, you could look to see if you're releasing all of these mosquitoes, how long are they persisting in the environment, what the ratio of your sterile mosquitoes are to your regular mosquitoes as well. So I think there are a lot of different applications, even in the mosquito field, that could benefit from this.
Okay, and I have to ask, species identification versus individual identification.
Okay, so this is kind of one of the biggest questions, and it's easier for sure to do species than we can do species for sure. Individual, it depends on a lot of factors, but one of the things we've been finding is that the DNA is more intact than we believed it to be. That means instead of short stretches of DNA, which it's hard to assign to genetic ancestry groups or hard to assign to individuals. But as we use these longer sequences, there's new technologies coming on stream again from the medical field where you can sequence long stretches of DNA. And actually these handheld nanopore devices that I showed you, they're designed to sequence long stretches of DNA. So it depends how long you can get, but if you can get this sequence long enough, that tells you that all of the DNA in that one individual sequence comes from one individual. So if you can get it long enough, you may have sufficient markers there that you might get to individual level. but it's something that's been pursued by the eDNA field because there's a lot of benefits to getting to individual level, or at least close to it. You can start to not know how much DNA is present, but actually potentially how many individuals that came from. You can do stock assessments then. So if you think about fisheries, which pioneers a lot of eDNA work, you can do a lot more with this technology. You can also potentially look at other regions that you might be interested in to maybe do sex ratios. So again, for sea turtles or fisheries or wherever your interest is, right now, if you want to know what the ratio of the sex ratio is or the age of the population, you have to get hands-on and capture those animals and do a lot of measurements. But there's indications from publications that actually, as we get better at doing eDNA, as we can increase the number of regions that we look at, you may be able to start incorporating some of those things like age profiles. Did your DNA come from a mass spawning event, or is it really mature animals that are reproducing that's coming from?
Well, lots of fertile ground for high school and college debate teams to be working with that. And I love that we got the kind of detail. Thank you. I'll hand it back to you in a minute for the final word, but to all of you who have joined us for this recording, whether it is the video or the podcast, we thank you for spending your time celebrating with us the people who are out there making new things happen to improve the lives of people near and far. We hope that you will tell others about our series, the hundreds and hundreds of videos that we have on our YouTube channel, talking to people who truly give us inspiration to get out there and to see how we can make differences as well. As we always like to do, we hand it back to the speaker for the final word. Dave, what would you like people to have in mind as they finish watching this video or listening to this podcast?
Yeah, I think what I would hope they would have in mind is just how much of a serious issue the biodiversity crisis is. It's going to take a lot of intentional effort and people's part, including volunteerism and including conscientious consumerism, to really help to address it. And I would say that environmental DNA offers this great way of measuring biodiversity and if we can restore biodiversity. But it really is boots on the ground, frontline conservation that's also needed to actually make the changes. So eDNA is going to be really powerful to make sure that we're making the right changes. But we do still obviously need people to be involved and to try and protect what we have while restoring what we've lost.
Excellent. Well, thank you. Thank everyone. We will see you next week.
Thanks.